Hello, everyone, and welcome to the COVID More Than 6 Feet Apart session. My name is Chelsea Spruance, and I am a 28-year-old adult with CF. I'll be moderating this session this evening, and I'll be introducing our other amazing speakers in just a moment. This session is also being recorded and will be available to all the registrants after the event. Before we get started with this session, I want to go over a few items related to the technology and how we can all interact. First, closed captioning is available and should be showing automatically on your screen. You can hide these or adjust them by viewing or how you view them by clicking the CC icon in the bottom of your screen. We will be taking audience questions during this session, so please submit your questions at any time to the Q&A box. We won't be able to answer questions about individual diagnoses or treatment plans, and we encourage you to use the information that you have today to have a conversation with your own specific care team. Otherwise, we will do our best to answer as many questions as possible verbally. And we also will be typing answers in the question box by typing them directly into that Q&A box under that question. So please check back there if you haven't heard your answer verbally to see if it's been responded to. This session is 75 minutes, and we will be leaving the last 15 minutes for question and answer. Now, we know 15 minutes is not a whole lot of time, and we anticipate a lot of questions coming in about this topic. So that is why this year we have a Saturday session specifically for question and answer. So if you saw or submitted a question you would like to hear answered that we did not get to today, please return on Saturday to hear us continue to address the community's questions. You'll also notice the group chat for everyone who is in this session. Please note you'll want to change that drop-down menu so that your messages go to panelists and attendees and not just one individual person. That way everyone in this session can see your comments. If you have a question for the speakers though, be sure to put that in the Q&A box to submit it, not the group chat. In this session, there will also be a few polling questions that will pop up from our panelists. You'll be prompted to answer those throughout the session. And these polling questions will give us an idea of who our audience is today and be able to interact with you throughout the session. This is also a time I would like to say that any personal stories shared in this session are to be taken as personal opinions, stories, and feelings. These stories should not be taken as medical advice or guidance for your own care or situation. And we encourage you to ask any questions regarding your own personal care directly to your care team. If you have any technology problems during the session, please return to the event homepage and click the live support chat icon at the bottom left corner of the webpage and connect with a tech and support program. Now, with all these logistics out of the way, I would love to invite our panelists to turn on their cameras so that they can introduce themselves to you. So, get off with the introductions. I'll start with myself. As I said, my name is Chelsea. I'm a 28-year-old adult with CF, joining you from the US Virgin Islands. I've been involved in many virtual conferences with CFF, but ResearchCon really excites me because it gives us an opportunity to see how many people are working so hard to better the lives of the CF community. Next up, I would love to introduce Christian.
Thank you very much, Chelsea. I think we would love to have ResearchCon in the Virgin Islands, maybe next year, if you'd invite us. Thanks very much for allowing me to be part of this. My name is Christian Merlo. I am an adult pulmonologist. I work at Johns Hopkins, and I take care of people with CF and those who may require lung transplants because of their lung disease. And I will pass the baton off to Yvette.
Thank you, Christian. My name is Yvette, and I am a CF spouse from Georgia. We've been together for 13 years, and I joined ResearchCon this year to just share our personal experience of what this last year has been like and some of the silver linings and not so great moments. Next up, we will hear from Natalie.
Hi, everyone. I'm Natalie West. I'm also an adult physician pulmonologist at Johns Hopkins. This is my first year presenting at ResearchCon, and I'm super excited not only to hear from other providers, but also in particular from CF families and people. And next up is Lael.
Hi, I'm Lael Yonker. I'm a pediatric pulmonologist and CF Center Director at Mass General Hospital in Boston. And this is my first ResearchCon as well. And I study airway inflammation in individuals with cystic fibrosis and collect specimens like blood and sputum from individuals with CF. And when COVID started, I started up a parallel biorepository collecting samples from children who were either exposed to or infected with COVID. And so I'll be sharing some of what I've learned over time as well. And next I'll pass it over to David.
Hey, my name is David Pruit. I'm from Dallas, Texas. I'm a CF patient. I'm also a ResearchCon first timer. And I chose to participate in ResearchCon this year in an effort to give a voice to the patients, and then hoping that my experience and my journey with COVID and CF can help other people.
And next up, we have Patrick.
Oh, Patrick, yeah, right.
I'm Patrick Flume. I'm also an adult pulmonologist at the Medical University of South Carolina in Charleston. I guess I'm the old guy on the crew. This is not my first rodeo. I have long been involved in clinical trials and clinical research, especially CF, but this last year has been devoted to COVID-related research, particularly the vaccine trials, and I'm looking forward to sharing an update on where we are with the vaccine studies. Chelsea.
Thank you, Patrick. Thank you to all of our amazing panelists for setting time aside from this crazy world that we've been living in to come speak to us today. So you'll notice our polls will pop up on the screen as we continue, like right there, and we will encourage you to answer them as they're posted to allow us to get to know you a little bit better. This session is truly unique because COVID-19 is something we all have experienced throughout the entire CF community together. It is no doubt that COVID-19 has had a significant impact on mental, physical, and the entire well-being of everyone. From people with CF to their families, to care providers, researchers, and care and research institutions as well. This session will consider these different viewpoints and cover topics including the impact of COVID-19 on the CF community, telehealth, and CF research. In this session, we will learn about the latest research, reflect on how CF has informed preventative measures for COVID-19, and highlight the successful path to vaccination. We encourage you to answer our next poll as we begin our session. As you learn from all of our introductions, we are joined by both clinicians as well as members of the CF community like myself. This means we will be hearing from both the clinical side of COVID-19 as well as personal accounts from people who know what it is like to live a CF life in a COVID world. To start our session, I would love to invite David to share his story with you. David?
Thank you, Chelsea. Okay, so I'm going to tell y'all my journey with COVID in 2020, and living with CF in conjunction with that. And so on October 26th, 2021, or sorry, October 26th, 2020, I got exposed to COVID-19. I obviously didn't know I was exposed. I was at a family event, and I'm pretty sure it was my dad. And so he kept feeling more sick and more sick, and I was just fine. I didn't have any problems or anything. And so then we all the whole family was go get tested, different testing sites here in Dallas. And he tested positive, I tested positive. So because there are 2 members in my biological family of origin that have cystic fibrosis, we had to get really serious really fast about, not getting. One of them had a double lung transplant, so he's immunocompromised already. And so we had to really watch it with, exposure. And everyone else tested negative. And one of the other siblings in my family is an anesthesiologist. And he recommended, like, hey, everybody, just get tested every 5 days for like the next, 20 or 30 days, just to make sure, you're still negative. And so everybody did that. And mine, the other family members decided to do COVID however they want to do COVID. And I decided to go quarantine in a hotel because I was living with my sister, and she has cystic fibrosis. So she kept testing negative. The double lung transplant guy kept testing negative, and I kept testing positive. So I actually had 10 positive tests, 5 negative tests, and 2 antibody tests from the span of October 30th, 2020 to November 20th, 2020. And I was mostly asymptomatic. I had a sore throat, and I didn't lose my sense of taste and smell. A lot of the stuff on the internet that they're putting out there, I didn't really have those kind of symptoms. My dad, on the other hand, he's 70. He ended up at UT Southwestern for 5 days. His thing got way more serious, way faster. And I think the reason why mine went so well is because I had started Trikafta in September of 2020, and my lung function went from 68% to 90% in 45 days. And then, over 120 days later, I went to my pulmonologist again, and I was over 100% lung function. And I still have over 100% lung function. So Trikafta made me COVID immune. I mean, for me, for my personal story, which was pretty incredible, really good timing for me. In terms of mental, psychological, social, aspects versus the physical, medical aspects, those 2 weeks I was quarantined in a hotel, I was diagnosed with COVID anxiety I went to an emergency room one day, and the doctor said, you have COVID anxiety, just because I was in social isolation, and I was really messed up in the head. I mean, I was, you can't talk to anybody, and I'm a very social person, so that's very difficult for me. And yes, that was the hardest part. I was asymptomatic, and since, I mean, the next 2 weeks were probably the hardest part. I had the whole emotional, psychological, isolation, and that was difficult in a different way than the last 2 weeks in November, like Thanksgiving and all that leading up to Christmas. I did have a lot of fatigue and exhaustion. So the first 2 weeks I was kind of in shock and anxiety, and the second 2 weeks I was in a lot of fatigue and exhaustion. And I have since then been vaccinated. I was put on the list as a 1B here in Dallas. So I got vaccinated with Pfizer on 2021, and then 2021. And vaccine went well. Yeah, I would highly recommend anybody who wants a vaccine, get a vaccine. And that is my COVID testimony. And if you have any other additional questions, I'm happy to answer them. And there you go, Chelsea. Thanks.
Well, that's definitely a good New Year's present. I didn't realize you got it on the first of the year. But no, I really love that you touched on the fact that you did develop COVID anxiety from not being social. You are muted, David. But I think that was really interesting to share, because I think whether we had COVID or not, that's something that we all experience in some sense of the way throughout this entire year, is that social isolation and feeling the mental effects of that. But thank you so much, David, and thank you for bringing the attention to your impact on with Trikafta on COVID as well. I personally
very welcome.
I had the flu, so I can
Okay.
right after getting on Trikafta, so I can understand that.
It's
And
amazing.
it definitely was very helpful. I'm glad to hear that that helped you in COVID as well. Do you feel any residual effects from
So
COVID,
I've tested with my
are you-
pulmonologist. I have no long-term damage, and I'm over 100% lung function. So it's incredible. I mean, I get a new lease on life. It's great. 2021 is looking good for me, you know, so far. Yeah.
Did your fatigue or anything continue after you stopped testing positive, or were there any like lasting side effects for a little bit longer?
It's just that I had the fatigue and exhaustion. A month later, I got a pretty big medical diagnosis that really rocked my world that I'm in right now. And so that caused a different level of stuff. But COVID wise, I mean, great. I mean, it took a month. It took me down for a month and I just processed through it. And then I got vaccinated and now I'm peachy. So
That is
Yeah, it's
amazing,
great.
David. I'm so happy that you were able to go through that and so well on the other side. Well, thank you so much for your testimony. And next, I will invite up Natalie West and Lael to respectively talk about COVID outcomes in people with CF.
Thank you so much, Chelsea, and thank you, David, for sharing your story. That was great. You had a lot of people in the chat thanking you for being so open and honest. And so in this first section, Lael and I are going to be talking about outcomes of COVID in people with CF and touching a little bit on CF research in COVID. We have a few disclosure slides that I'll let you read through yourself. And so you just heard David's personal story about his COVID diagnosis and his experience as well as vaccination. And next we're going to talk about COVID outcomes on people with CF. I'm going to focus on the adult outcomes as I'm an adult pulmonologist, and Lael is going to focus on the pediatric outcomes. And then we have a few moments to talk about COVID research and what the CF Foundation is doing to support research. So the best news is that people with CF infected with COVID are doing way better than we expected. I think everyone on this call remembers how scared we were when COVID first hit and really not knowing how it was going to affect the world, but also the CF community. And so the first study that was published on outcomes in people with CF was published in the Journal of CF back in November. And this was really a global effort. And data came from 19 countries spanning 6 continents and reported on 181 people that had been diagnosed with COVID prior to June 13th of last year. And the great news is that outcomes were very similar to the general population. And so there were 11 people that had to be admitted to the intensive care unit. 7 of those were post-lung transplant. And unfortunately, there were 7 deaths at that time. Now, any death is just purely not acceptable. We don't want any deaths from COVID. However, it was much better than we initially feared. And what this group looked at was, are there any risk factors that would help predict more severe illness, meaning hospitalization or death? And there were 4 main risk factors that kind of panned out. One is older age, which is not surprising. Those that had lower lung function, so FEV1 less than 40%, were at higher risk of a worse outcome. People who had CF-related diabetes, and those with organ transplant. And there was a question in the chat, the first question that came through the Q&A was perhaps why was there a lower death rate? And so we're going to talk about that a little more, but this particular group hypothesized that the younger age of the general CF population might be benefiting us all. And I want to point the European Cystic Fibrosis Society, they have a website. So if you look over here at the bottom right, there's the link to it. You can go to it at any time. And there are 27 countries who are frequently updating a registry, and all the countries that are participating are shown here in the map. And they enter data on all COVID CF infections, and so that the European Cystic Fibrosis Society can follow outcomes. And what you'll see on the main page is the summary. It looks something like this. And it was last updated March 8th. I did check this morning. And it shows a summary of what the cases look like. So To date, as of March 8th, there was a little bit over 1,100 cases reported from 27 countries. Pretty equal gender. And then we'll dive into a little bit more of severity, but the majority are mild or asymptomatic. Unfortunately, to date, 13 people have died, but 816 have recovered. And so on this main site, they also show a couple graphs that I think are interesting to look at. So they look at the percent predicted FEV1 by category. So FEV1 greater than 70% is considered mild CF lung disease. You can see the majority of infections were in this category, which is not surprising because the majority of people with CF do have mild lung disease. 41 to 70% are considered moderate lung disease and less than 40% more than severe. And then looking at the severity of COVID-19 infections, it's reassuring that the vast majority are mild or asymptomatic, meaning no symptoms. There were 19 that were severe, which that means they have a high respiratory rate, they're short of breath, their oxygen levels were low, or they had abnormal x-rays. And then there were 7 that were critical that needed ICU level care. And then looking at outcomes, again, here is a graph representing the vast majority that recover. So what about here in the US? So it's nice, the CF Foundation Patient Registry sends all the care centers a weekly email with the updated numbers. So we just got the email this morning for this week. So all these numbers are fresh, hot off the press. And there is 1,383 cases. 71% of those are adults and 29% are pediatric. And you'll see that about 1 in 5, so almost 20% of adults need hospitalization. And for pediatric children, 1 in 10 need hospitalization. 7% of these almost 1,400 are people who have advanced lung disease, so FEV1 less than 40%, and 7% are post-transplant, majority being a lung transplant. To date, unfortunately, 14 people have died. 8 of those were post-lung transplant and 3 were in that category of more advanced lung disease. And what we were seeing is there is racial disparities in COVID infections in CF. So what you may have heard nationally is that the African-American population and the Hispanic population are more often and disproportionately affected with COVID and also have worse outcomes. So to remind you the demographics of people with CF, 93% are white, almost 5% are African-American, and about 9% are Hispanic. And when you look at the percentage of COVID infections, you'll see at this bottom line, Hispanic has 13% of the COVID infections, but they only count for 9% of the population. So what we're seeing on the general population is holding true for those with CF. So I'll pass this on to Lael to talk about the children outcomes.
Thank you, Natalie. So as I mentioned before, I'm a CF doctor, pediatric pulmonologist, and study airway inflammation. And so when COVID started, we all, it's a respiratory illness. And so I started up this biorepository to study how COVID affected kids. Next slide. And early on, you can see in the red box, we all, we thought kids were spared from COVID. And that was probably because we were testing kids because we didn't know that their presentation is so different. And also kids, when quarantine happened, kids were kept home from school, and as opposed to the adults who had to go to the grocery store and sort of interact in some level of the world. And so, so the kids were more protected. But as things have opened up, and now, kids are back and have following their normal routines, and young adults as well, we can see that children, these are numbers for Massachusetts over 2-week increments, and you can see that children ages 0 to 19 and 20 to 29 have the highest rates of COVID positivity. And this is seen in many places. Next slide. But fortunately, as Natalie pointed out earlier, the mortality is significantly lower in this age group. So you can see on the left is the reporting of the cases, and I think that the under 18-year-old is underrepresented here on the left in the cases column graph or bar graph, but on the right you can see the deaths, and the top one under 18, 0.2% death rate as compared to older adults. Next slide. And one more, just advance one more. But the important take-home message is that children, it doesn't matter how old you are, you can carry the same amount of virus in your airways. Natalie, if you just hit one button to show that. So you can see across the age range from newborn through a 20-something, you have the potential to carry just as much virus in your airway. So so you're just as infectious if you're young versus old. The difference is that the virus decreases over time. So early on in the infection, especially while you're just caught the infection, or in the early asymptomatic period, or just starting to develop symptoms and thinking if this could be COVID or not, that's when you're most infectious. And I think we all know that at this point. It's seen in kids and in adults. These curves will change in vaccinated individuals, but vaccinated individuals can still have virus in their airways, and children are not vaccinated. So under 16 are not eligible for vaccine currently. And so this is something to be mindful of. Next slide. And we know this, we've lived this, we've seen this. Kids have very mild symptoms when they're infected with COVID. And this holds true with CF as well. Very mild, often asymptomatic. And you can see COVID negative. So people who thought maybe they had COVID, but were tested negative, people who tested positive for COVID. And I'm going to talk about this third sort of presentation, a late presentation of COVID. But in the kids who were COVID positive or who had just a regular old cold, you can see there's no difference in symptoms. And these symptoms are symptoms that children and individuals with cystic fibrosis are very familiar with: cough, congestion, runny nose. So very hard to tell when this is COVID or not. So it's tricky, makes things complicated. Next slide. And this is that third scenario that I mentioned: multi-system inflammatory syndrome in children, or MIS-C. And this is a delayed complication of COVID in kids. There is a version in young adults as well, MIS-A for adults. And this is a complication that occurs about a month after you've had COVID, or a child's had COVID. And they may not have even known that they had COVID. They may have been asymptomatic when they were first infected. And then about 4 to 6 weeks later, they develop fever, maybe rash, maybe GI symptoms like diarrhea or vomiting or just abdominal pain. And then they can have cardiac involvement, and they can become quite sick, and their heart could have aneurysms or severe complications. And you can see this is rare compared to the amount of COVID cases amongst kids, but it's rising. And the COVID cases are rising. So a certain percentage are going to get MIS-C, and the more kids who have COVID, the more MIS-C cases we'll see. And this was from early March. It was about 2,800. And I just looked this morning, the reporting was just at the end of March, so they're still not up to date, but it's about 3,200. So the rates are going up, and it's something to be mindful of. And I think it's just a reminder that just because kids don't have significant symptoms to COVID when they're initially infected, we want to avoid infection for kids, because there's this possibility of a more severe risk, and they're also contagious, and we want to avoid the continued spread of COVID. Next slide. And I just want to touch on COVID and pregnancy, because we also followed, we are following babies who are born to COVID positive moms. And so if a mom gets sick in pregnancy, that's very hard on the mother. Mothers tend to, and certainly mothers with cystic fibrosis, if you were to get sick, that can have significant effect on your body. It can make the pregnancy riskier. And there's also a concern that if you had COVID, you could have, like other illnesses with high fever, you can have negative outcomes on the child. So we want to avoid COVID during pregnancy. And as an upside, though, some of the antibodies that you have do get transferred for the baby, but not very high amounts during the actual, if you're just infected naturally. If you're vaccinated, there's a few benefits. First, the pregnancy is safe the pregnancy, it's much better to not have COVID. So you want to avoid COVID. So you're vaccinated, you avoid getting COVID during your pregnancy. And there seemed to be more transfer of antibodies to the babies. So maybe the babies are better protected. I would still recommend, wearing a mask to keep the babies healthy. But because of these reasons, vaccination is safe and recommended for pregnancy. Next slide. So my sort of overall advice, my sort of public service announcement is, please continue to wear a mask. I think you all, the CF community knows masks well and wears them well. But just to remember that children are still vulnerable to infection. They're not eligible for vaccination yet. Vaccinated individuals could carry the virus and pass it along to unvaccinated individuals. So continue to protect kids, wash your hands, social distance, and when vaccines do come along, please do get them. Thank you. Oh, next slide. So now, Natalie and I are going to talk about the effect of COVID on research. Next slide. So COVID has affected CF research in many ways. First, when there was the big shutdown, there was essentially a shutdown in all research except for COVID. So COVID research was prioritized, everything else was paused. People weren't allowed in labs, and people weren't allowed in hospitals. People weren't able to come in for their clinical trials that they were actively participating in. So really the clinical trials shut down as well, or had to adjust or stop early. And then when they did start to open up, you have to go through COVID testing. So it makes, to come in, and different hospitals have different policies, but it's a whole nother level of complication that impacts the ability to do research easily. But as a positive, the Cystic Fibrosis Foundation has invested in understanding how COVID affects the CF community. And we were worried initially that CF patients were going to have a hard time with COVID, but then the numbers were better. So why? What is it about the CF airway, and I think, and Natalie's going to talk about some of the areas that we're looking into, but there's actually a lot that I think we're going to be able to learn.
Great, thanks, Lael. And so yes, the CF Foundation has awarded $2.7 million as of December for 11 studies looking at CF and COVID. And they all fall into 4 different categories. The first is the impact of CFTR modulators on inflammation and mucus. Is Trikafta, Symdeko, or Cambi, or Kalydeco affecting how much inflammation occurs during a COVID infection? The next category are cells from people with CF more or less likely to become infected? And how do the CFTR modulators affect that? The third category is whether COVID leads to increased bacterial infection. Sometimes in other viruses, for instance, the flu, we will see an increase in Pseudomonas or staph needing antibiotic treatment. And lastly, are antiviral therapies effective in CF, just like they are in the general population? And then the fourth category is, do cells from people with CF have an altered inflammatory response? So investigators are comparing cells from people with CF to cells from people that do not have CF and looking at the inflammatory response. And so next, we'll turn it over to Chelsea and Patrick to talk about COVID vaccination and reopening.
Thank you so much, Natalie. I really enjoyed the fact that both of you highlighted the fact that even though the mortality and the severity of symptoms is rather mild or manageable in both people with CF as well as adolescents or children, it's still so important to continue masking, continue social distancing, and continue being as vigilant as it has been for the past year with COVID-19. It's been a long year for all of us, but it is a good reminder that even though these are lower death rates, lower mortality rates, lower symptom severities, it's so important to keep our vigilance, keep up that effort that we've been doing for the past year. So thank you for that. And thank you so much, David, for sharing your amazing story with us of overcoming COVID yourself. As we move on to our next topic about vaccines, you will see a poll popped up. And thank you so much for answering that as we begin to dive into vaccines. I would also like to inform our audience that we discuss the vaccine as we start to discuss that vaccine. We will not be addressing the rollout of the vaccine because that is an issue that varies state to state and situation to situation. So please refrain from adding any questions about vaccine rollout or availability to the Q&A box, and instead reach out to your own personal CF center. As we move into the vaccine, I myself am fully vaccinated, and I know I was one of those people that was slightly worried about getting the vaccine. I just was unsure. I didn't do enough research on my own. So in order to ease my mental worry, I reached out to both my care team, my support system, people that I knew who had reliable information, like my CF center, like doctors and nurses, and as well as my family around me, to see what the best educated decision was for me and the best way that I could educate myself to feel comfortable about getting the vaccine. And ultimately, thanks to all of that really great information that I received, I felt more than comfortable getting the vaccine. And my vaccine was perfectly fine. The second vaccine took me out for a good little bit, for about a day. It was a good Netflix and couch day. But other than that, I had a fine experience, and it really does give me peace of mind now. Not that I'm going to lessen any of my strategy around avoiding COVID, but it definitely does give me peace of mind to be able to go about more safely through the world around me. More importantly, I know why you're here, is to talk to Patrick, our next speaker, about the COVID vaccine. So Patrick, would you like to come on and share?
Thank you, Chelsea. First, just my disclosures. Included in there are studies with vaccines. We have worked with 3 different companies in their development of the vaccine. So we'll start talking about, just give you an update on the COVID vaccines. And I want to approach this subject by giving you a little bit of background on how the immune system works to fight infection, but this is going to be a very abbreviated version. Describe how the vaccines work, how those studies are conducted, and then when they are approved, or more appropriately authorized, what does that really mean? And then try to highlight what I think would probably be some of the key unanswered questions, although I'm sure there will be plenty more. So I'm focusing on the antibody response of the immune system, and these antibodies are highly specific for what we call antigens. These antigens are these molecular structures that are sticking out from the pathogen. The antibody forms to that and is very highly specific, almost like a key to a lock. So you see these cartoon examples for measles and chickenpox, how those antibodies are very specific for those antigens extruding out from the portion of the virus. How these antibodies are made, first part of your immune system addresses these pathogens that have entered you, and these are your macrophages. These are important white blood cells that process the pathogen itself, but then looking at those proteins, because it's going to present them to other cells. And in this case, they're going to present those antigens to the lymphocytes. And there are different types of lymphocytes. The B cells are those lymphocytes that will create the antibodies. The T cells actually can help stimulate those B cells to do their job. And those B cells will then replicate. They'll make copies of themselves to be able to make these clones turn into plasma cells, because these can reside in the body and be that memory, if you will, for when you ever get exposed to that again. And they're producing those antibodies to target those. They're circulating, prepared for those pathogens if they come into you. So the vaccine, the intent is to show that antigen, that protein, to the immune system. And there are different ways that that can be done. The first, the top left, is actually just give the virus to the person. Now, generally speaking, that you could kill that virus, you could inactivate it, or you could give a live version of that virus, but you would weaken it, just attenuate it. This is not the way that we are doing the vaccines for SARS-CoV-2 to try and prevent COVID. A second approach is to take other viruses and put in the information about the protein into that virus, so that virus can come in and harness the power of the cells to crank out this protein, and that's what they can respond to. And so you can use different types of viruses, those that replicate or those that won't replicate. And that's what we've been using the AstraZeneca model, Johnson & Johnson, Janssen, which is the same as the Johnson & Johnson, are using a non-replicating, non-human infecting virus to deliver it. The next mode is to actually give the information through either DNA or RNA. So you'll hear about the messenger RNA versions. You're basically taking the information to code for that protein, give it to the cell, harness its power to crank out that protein. And that is the method that is used by the Pfizer and Moderna model. And although this seems like the newest kid on the block, actually these have been in development for over 20 years. So when people think about these moving too fast, it's actually been, people have been working on it for a very long time. The final approach is actually just give the protein to the person. You could synthesize that protein, you could create it by recombinant methods, package it, and then deliver it to the cells. And that is the method by which Novavax is trying to deliver that protein to which they would make antibodies. So here is a drawing, a rendition of the SARS-CoV-2 virus, and you can see lots of different proteins sticking out from that coat. Those are all different targets by which antibodies can be made. And the target that is typically used for these vaccines are the spike protein. You see those big pink or purple globs sticking out of the top of the virus. So how vaccine studies are conducted: once you have a vaccine that you can give to people, you need to go to areas where there actually is risk of infection. If you go to Australia where nobody has COVID, you're not going to be able to pick up cases. So you want to go into areas that are endemic. There's a lot of infection happening. Turns out that was pretty much all over the United States that this study could be conducted. You then need to recruit large numbers of subjects. And so these studies are typically 30,000 in number. The inclusion criteria are generally healthy, medically stable, at the beginning, adult subjects. And those that were excluded were those that had already had a COVID infection. They have problems with their immune system or had been given recently some blood products, because you're going to measure antibodies and you don't want them to be the antibodies of someone else. And then the key endpoints that you look at, you are always looking at safety, same as we are for all of our clinical trials. So you're looking at the adverse events. But the primary endpoint of these vaccine studies is looking at prevention of symptomatic COVID illness. And that's terribly important. It's not preventing COVID or preventing SARS-CoV-2 infections. It's actually preventing symptomatic infections. But you can actually look to see if it prevents infection overall. So the vaccine study, by design, they're all pretty similar in this sense. You get consent and there's screening. There is a day one. These studies are typically 2 years in length that we're going to monitor these subjects. And so they will get their first shot at that first. Oftentimes they're screened and dosed on the first day. Many of our studies are 2 injections. So I've shown here an example of one in which the vaccine or the placebo was given 4 weeks after the first dose. There will be a plan for how you monitor these people over time. In general, if these people are doing well, you're just bringing them back, looking for adverse events, measuring antibody levels. But when you're looking at the clinical endpoint, you're looking at symptoms that are occurring starting 2 weeks after they have been fully vaccinated. You certainly are monitoring the entire time, but for the important endpoint, that 2-week period is what you're looking at. So here's an example of one of those. These are the data from the Moderna study. And you can see at the bottom left, that first arrow was the first dose. And then at day 29, that second dose. But you don't really start counting cases in terms of your primary endpoint until 2 weeks after that point. So that's that dashed line. Then what you do is you wait for cases to occur. And when a sufficient number of cases have occurred, You break the blind to see who got vaccine and who got the placebo to see if there was benefit. And as you can see in this graphic, there was obvious benefit occurring actually pretty early on after the dosing of the vaccine, which led to the authorization of the Moderna vaccine. Keep in mind, this did not require a large number of cases. Many of these studies may only rely upon the first 150 or so cases before they can demonstrate that clinical benefit. We also look at safety. These are data from the Moderna trial looking at safety. So you can look at adverse events that occur locally at the site of injection, which we're showing on this one, comparing the side effects that occur after each of the doses. What you can see on the left of the panel is that a large proportion of the people who got the active vaccine had some adverse event locally. What you also can see is that for the most part, these are very mild. When we talk about grade 1 adverse events, these are very mild events. And so when you look at the specific events, whether they're pain or redness or swelling, where those occur, mostly they were pain, and again, mostly in mild, and they were also transient. When you look at systemic side effects, you can see that even in the placebo group, there was a high proportion of subjects who reported systemic events, and they got just a shot. But when you look at those that in the Moderna study, there was a greater proportion of people who had complained about side effects after the second injection compared to the first. But when you look at what those might be, whether it's fever or headache or fatigue or muscle aches, again, most of them were mild. And again, most of them were very transient. So in general, these have been considered highly safe. Now we talk about the approval process, and these were authorized for use under an emergency use authorization, or an EUA. This is different than a typical FDA approval like may have been used for a drug like Trikafta. To get a EUA, you need to be talking about a serious or life-threatening disease or condition. You have to have evidence that it is effective. You have to look at that risk-benefit analysis, so comparing those side effects that we see, and there has to be no alternative. And those all are clearly met in a COVID situation. The EUAs are effective until the emergency declaration ends. So if some point in the future, the federal government decides that we are no longer under an emergency declaration, then these companies will have to appeal to the FDA to get final approval to continue distribution of the vaccines. So I just want to cover a few questions that I think are likely to be considered. Which vaccine should I get? Do I need a booster injection? A little bit about the variants, and then also, does it actually prevent infection and contagiousness? So which vaccine should I get? This is actually quite difficult because these vaccines were not compared to each other. And so although it's tempting when you hear numbers about efficacy, 90%, 95%, 65%, and think that those are quite different, we don't know that to be sure. And actually, when you really get to look at the details of how the studies were conducted, grossly they seem to be very similar, but there actually are some very important differences. And we can't assume that they all were being studied in the same level of risk of infection. So be cautious about reading the data to try and figure out which one might be better. What we do know to be true is that these vaccines substantially reduce the risk of symptomatic COVID illness, and they substantially reduce the risk of hospitalization for COVID illness. So the purpose of the vaccine is to protect ourselves. The final point is, for the most part, we all won't get a choice. And if you think about when you got your influenza vaccine, if I asked you which one did you get, you probably didn't know that there were differences between the influenza vaccines that were available. So the conclusion there is, get the vaccine that you can get. There are some data that we have through the registry about vaccination rates in persons with CF, and as of April 9th, we were up over 11% of eligible people with CF. These data are likely to fluctuate. For most states, people with CF weren't eligible for the vaccine until just recently. So we are hopeful we'll see those numbers rise, similar to what we see for our influenza vaccine rates. Is there benefit after the first injection? So most of these vaccines, you're getting 2 shots, but we can actually look at the data to what happens after you get that first injection. And what you typically see is that after about 10 days is when you begin to see separation of the infection rates in those who got placebo compared to the vaccine. So it is evident that there is some protection even after that first injection. And although it's tempting to think, well, maybe I only need the 1 injection, the advice is the drugs were studied with 2 shots. That's what we're confident in, get the 2 shots. And so follow the recommendations for how they're proposed. What about the variants? We hear a lot about these variants, essentially that as these viruses, like any other collection of DNA, keep making copies of themselves, there is the potential for mutation that makes them somewhat different. That doesn't always mean that they are going to be something bad. We could have a variant that occurs that no longer makes the virus contagious. But the more that this virus is able to replicate, the more opportunity there is for variation between those viruses. So there will always be variants, but there will be more if we don't reduce the infection rate. So when we have a forest fire with these infections, we have to anticipate there'll be greater number of variants appearing. You will hear that some of these variants have greater infectivity, meaning that you may not need quite as many viruses to increase the chance that you would become infected. You will also hear that there is a suggestion that the antibodies that are developed, whether one has infection or gets a vaccine, are less effective. Now, this doesn't mean they were studied to see if the vaccines prevented the infection. This is looking at the antibody levels that are necessary to neutralize that particular variant. And so in the laboratory, you can say that there are differences. That does not mean that you are not protected. And so the early evidence that we have is still suggesting that the vaccines are protective against the variants that are currently circulating in the US. So the conclusion there is get the vaccine. Now to the more complicated question is, does the vaccine actually prevent infection? So how can you assess the ability to prevent infection? There are a number of ways that this can be done. If you wanted, you could swab people every day. There's some limited sensitivity if they're doing it themselves. You can look at their antibody levels to try and see if there were some asymptomatic infections. There are some problems with that. There actually has been a proposed study that is likely to happen, and that is purposely infecting healthy individuals after they got vaccine or placebo and testing them to see what happens. That's a complicated study. I'm not sure I'd be willing to volunteer for something like that. Nonetheless, there is some evidence that suggests that there may be some reduction in actual infection. But the real question is, if you had the vaccine and you were to get infected, are you contagious? So what makes a virus contagious, how this virus is transmitted, has to do with how big that viral load is. If you cough, how much virus is in there. The tropism, that's the ability of the virus to infect the host. And then what's the actual exposure? How long were you with them? How close were you with them? And were you using any kind of filter like a mask? So what we do know is that there is an impact of the vaccine on the viral load. And the way that we look at viruses is we use a technique called polymerase chain reaction, or PCR, to detect the virus. And from that, we can estimate how many viruses are in that. What's the viral load? So what PCR is able to do is to make copies of very small amounts of the DNA. So it's capable of finding a needle in a haystack. If there's very little of something there, it can amplify them, but it amplifies them one cycle after another until it can be detected. And so you'll hear this term called cycle threshold, and that means how many times did you have to run the cycle before you could detect the virus? So if you can do it with a very few cycles, that means there was a lot more virus there. If your cycle threshold is much greater, that means you had a lot less virus available. So what you can see here, looking at these data, is that as you look at the number of days after that first vaccine dose, the cycle threshold seems to increase after you get past day 10. So you go from a cycle threshold of 25 to 27. That's a 4-fold reduction in the amount of virus. Does that mean you're not contagious? Well, this is a complicated slide, and I only want to walk you through a little bit as we go along the x-axis on the bottom. That's the cycle threshold. We're increasing. So as we're going to a bigger number, that means fewer viruses. The curve at the top is looking at how likely were you able to culture the virus from there. So with a cycle threshold of 25, which is where it was at the beginning, 70% of those would become infected. But when you get it to 27, that number drops to around 50% or less. So you clearly are reducing the viral load, which means you're making yourself less contagious. Doesn't mean you're 0% contagious. You've got to think about that exposure factor. And I bring you back to the masks. This is looking at who is wearing the mask, whether it's the receiver, the spreader, or both, and what kind of mask you're using, what percent of reduction can you do? And if you put the mask on the person who has the infection, you can reduce the risk of passing that by about 70%. So getting the vaccine, wearing a mask makes a huge difference. So I'm going to conclude. Get yourselves and your CF team vaccinated. These are very effective. They reduce symptomatic illness. They reduce hospitalization. They reduce death. Vaccines may also reduce the risk of viral transmission. They do so by reducing your risk of getting infected and reducing that viral load. The safety profile is excellent. Adverse events are mostly mild and short-lived, and keep wearing the masks until we get the infection rate down. And with that, I'll hand it back to Chelsea.
Thank you so much, Patrick. Now I'm just sitting here wondering if my reaction to the vaccine was a placebo or an actual reaction, since they both showed reactions. But again, thank you so much for all of that information. I feel like it was something that was very, very wanted and very, very helpful to help inform everyone about something that maybe they did not know much about. So moving right along, we're going to go to our next topic, and one that we have become very familiar with this past year, telehealth. So again, we would love for you to answer the poll that popped up about telehealth. And with that, I would love to pass it right over to Yvette so she can share her story. My mouth does not want to work. Her story about COVID and telehealth with her husband. Yvette?
Thank you, Chelsea. I'm Yvette again, and we are here in Georgia. My husband is 39 with CF. He is on Trikafta, but he does have really low lung function and is on full-time oxygen. So we tend to be pretty careful anyway. So starting in January of last year, some people in his office, he does work full-time still, just seemed like they weren't feeling well, you know, a lot of the winter stuff that goes around. So he had already started working from home full-time before the pandemic was really a thing here in the United States, especially in Georgia. I don't think we had any cases until the beginning of March. Once we did start having cases, we talked it over and we found it best for me to switch jobs just to keep him safe. I'm working privately for a family right now instead of the teaching role that I had before. And that kind of sent us into full-on quarantine where we are still living in. It's been pretty hard with isolation. We don't go into stores. He goes nowhere because of his oxygen compressor. He doesn't use tanks. So it's just sucking up all that room air and pumping it right in through his cannula under the mask. We do pick up orders for everything. We don't go to family gatherings. We haven't eaten at a restaurant since our anniversary in the beginning of March of last year. And so the pandemic has come with a lot of isolation, and a lot of sacrifices, changing what I did for work. And I even changed my plans just a little bit for school and things like that to keep him safe. And it's 110% worth it. I do it all again. But with that isolation and with kind of the shift of just trying to protect him, it definitely takes an emotional and like mental health toll on us both. He hasn't been able to hug another person other than me in over a year, like not his mom or his dad or his siblings or any of our nieces and nephews. And to keep him safe, I don't interact that way with other people either. And we love each other to death. We've been together for 13 years, but a year locked inside together, they always say too much of a good thing. So it's definitely been really trying, missing out as life goes on. Not everybody needed to be as careful or wanted to be as careful or wanted to be careful at all, thought things were blown out of proportion. So, we've missed life events, we've missed weddings, we've missed birthdays, anniversaries, funerals, things like that. One thing that's been pretty difficult with that is managing those family expectations. Not everybody has the same level of medical literacy. Since I'm going to school for nursing, understanding research is really important to me. I did a lot of my own research on how COVID spreads, how it doesn't spread, likelihood of getting it, signs and symptoms, as well as, information on, the vaccines. Are they safe? What is an mRNA vaccine? We did all that research, but not everybody does all of that. So there's, for instance, we're both fully vaccinated now. But this fall, there's a wedding for his cousin, and the family wants to know why we're not coming when we're vaccinated. And it's like, well, not everybody there will be vaccinated, and some of that is by choice. But there's also going to be plenty of children there, and they're not eligible for a vaccine yet. Their age group isn't approved, and it's just not worth the risk. In our opinions. And even his doctor has said, you guys can start visiting with people, but only in small groups, only with other vaccinated people, fully vaccinated, waiting the period of time that whichever vaccine they got says to wait. I believe we were told 2 weeks after our second dose. And so it's just really important for us to keep him safe. And not everybody understands that we can't get together for Mother's Day. We can't get together for a wedding, because it's more important to keep him healthy for a year or two so that he has many more years versus risk, him getting really sick this year. Another impact we've had is, on his health. Yeah, he's home in quarantine and safe and not around other germs, but what is that going to mean when we can go back to whatever the new normal will be? Will he be more likely to get sick because, he hasn't been exposed to germs and things like that? If he was sick right now, would there even be a bed in the hospital for him? And with such long waits to get things like PICC lines, we just did oral antibiotics if he wasn't feeling well or wasn't doing well. We've utilized telehealth a lot this last year, and that actually has been kind of an amazing thing. We are a little over an hour from our clinic, and so not having to make that drive, not having to adjust his treatment schedule in order to fit 2 hours of treatments in before he drives an hour to clinic, and then by the time he gets there, he needs a bronchodilator again. And that's been really helpful. We miss a lot less work. It always took 5 hours to do to see everybody, and now it takes maybe an hour and a half. It's really been beneficial not to miss the work. It's easier to schedule. And it allows a lot more check-ins. Like he can go every 6 weeks if he feels like he needs to. And and while there are a lot of positives, there's some drawbacks too. He can't give a sputum culture from home. You know, I know how to assess him and give some vitals to them, but it's not the same as his pulmonologist actually putting his eyes on him. So there's no hands-on, you know, there's less accurate PFTs and things like that. But when he's healthy, I think it's an amazing option. And I really hope it stays around. So with that, just kind of the pros and cons of what we liked about it, I am going to pass the torch back to Chelsea. Chelsea.
Thank you so much, Yvette, for your wonderful story. We're going to pass it right over to Christian Merlo to talk about telehealth.
Well, thanks so much, Chelsea, and thank you so much, Yvette, for sharing your story. I think all of us are very, very experienced now with telehealth, and so we're just going to spend a brief bit of time talking about the CF clinical experience in telehealth, given this pandemic. I just have a few disclosures, to not as many as Patrick Flume, so you won't spend as much time reading that, and I'll move on. The overview that I'll talk about is our experience prior to COVID-19. It's very, very, very limited. There are some potential benefits that Yvette highlighted, and we'll talk about some potential pitfalls. And then what are we going to do in the future? What are the hybrid models that we're going to go through? And so I started out with doing a quick literature search, and I just typed into PubMed telemedicine, and there's been an explosion in research over the last 10 years. There were about 1,000 papers in 2010, and that was 6 or 7 times more papers looking at how telehealth can affect outcomes in patients with disease in the literature. So why do we do this? I think in general, patients are satisfied. That technology has become easier and easier and easier to use. Oftentimes it's easier to communicate between patients and providers. I may see the inside of a patient's house. I may meet their dog. I may meet a relative that I wouldn't necessarily meet, which may help with that bonding process. And that can improve access to care. Patients, as Yvette said, don't have to travel. They don't have to take time off from school, from work. And this can help improve both the waiting time that patients are waiting to see providers, and also if that communication and that bond grows, that may help improve adherence. And then finally, as in COVID-19, telehealth is there to provide care in extraordinary times. So before the COVID pandemic, telehealth was utilized in CF in very limited settings. And mainly those were among folks who lived far from regional centers, where it was really difficult to fly there, very expensive. It took a lot of time away from either school or work. And we know, just as Yvette highlighted, there are so many advantages. Now, there are some disadvantages, but there are so many advantages to telehealth, especially during this last year. Reduced time off from work, reduced time from school, reduced expenses from travel. But we really had no idea about the patient or provider experience. And how do we feel about it? We thought, well, we should do this, but do we really know how patients are going to like or dislike this? Are we really going to know how providers and team and care centers are going to like or not like this? Well, this was a study published this year by Davis and colleagues, and this was a cross-sectional survey that was sent out to care centers across the country. And it was conducted at about 11 centers in the United States. And what you can see here is that the vast majority of patients had a high level of satisfaction, almost 75% of adults and caregivers for PEDS patients had a high level of satisfaction. And the most important thing here, if you could just go back there, one slide, I'm not controlling this, but the most important thing is most people had no experience with telehealth. So folks liked it and had no experience in the past. Now we can go on. There were some concerns with this, and I had concerns as a clinician, like how are we going to monitor lung function? How are we going to monitor people's infections? The Cystic Fibrosis Foundation has been fantastic in their quest to supply spirometers to patients with CF throughout the country. And so there's some concern among patients in the country about missing data, but most were not that concerned with missing pulmonary function tests or missing sputum cultures. And most people with CF and most caregivers of patients with CF were interested in future telehealth visits. You can see here on the left hand of the slide, those are pediatric folks, and the vast majority of caregivers were very, very interested in having some sort of hybrid model, in-patient visits versus telehealth. And similarly among adults, this middle pie chart shows that most patients are interested in some sort of model where they can come in sometimes and they can be seen by telehealth others. And the vast majority of caregivers who are taking care of pediatric folks and the vast majorities of adults with CF were very, very interested in home spirometry. Now, similarly, this is a parallel study by the Cystic Fibrosis Foundation led by Bruce Marshall. And almost 100%, I mean 97% of programs were providing, by August 2020, telehealth services. And this is crazy. At Hopkins, we were tasked with the idea prior to COVID to take 2 years to develop our telemedicine service for some of our chronic diseases within pulmonary and pulmonary care. Overnight, we went from 0% to 100% because of COVID-19. But it's not a perfect system. And I really think we need to step back here and realize that not everyone has the access that others do. And this highlights a discrepancy even within cystic fibrosis care, that some have no access to the internet, some have language barriers where telemedicine is difficult, some have not enough money to make this happen. And so I think it's very, very important that we realize that, yes, this is a very nice tool for most, but we need to remember not to leave anyone behind. And I think the highlights here, the greatest benefits that patients and families felt in this CF study were really savings of time and money to travel to the clinic and not having to take time off from work or school. Now, we also need to step back and get introspective and ask patients and families how they felt about their self-care. And it's very, very striking that the majority of patients and families thought that their self-care stayed the same. On the right-hand side, you can see that the percentages of patients who had home spirometers, scales, and pulse oximeters at the time of this survey. Now, I imagine that these numbers are much higher now as I can speak for our center that our home spirometer percentages have increased over the course of time. And then finally, would we recommend this to other caregivers who take care of patients with other chronic diseases? And about a third would recommend, highly recommend, a third of CF programs would highly recommend telehealth services to their peers. We're trying to figure this out, and we've tried to do this in a very, very, very fast pace. So I imagine that over time, we're going to get better at this. And it may be that we're not going to just do telehealth. We're going to have to figure out some sort of hybrid model between remote monitoring and in-person visiting. And then finally, I really want to highlight, this is our team. And I miss them dearly. I miss being around them. I miss the communication. For patients out there, we miss you. For healthcare providers out there, we miss you. For colleagues out there, we miss you. And the final thing I want to end with is something that I usually say to patients at visits. And I think that for people with CF and for caregivers of people with CF, you've been living with this 6-foot rule for decades. We've been living with it for a year. And I think if more people followed what you do in trying to keep healthy, this world would not be in the state it is right now.
Thank you so much, Christian. I know I am all for coming back to in-person meetings, especially down here. So you can come down anytime and we can do a clinic right here. Go to the beach. But
Chelsea,
Natalie, you
Chelsea,
can.
one last thing I wanted to just say. I saw your and I just wanted to say that he should be very, very proud for your leadership in heading up this ResearchCon session. I know we are.
You don't need to make him any more proud, believe me. Thank you, Christian. Natalie, you can click to the next slide. We'll post that in the chat. But unfortunately, it is time to end our session. I know we were not able to get to the Q&A tonight, but I hope you gained a wealth of knowledge. And please remember, we have our hour-long COVID-19 and telehealth Q&A session on Saturday, April 17th. It's at 4 p.m. Eastern Standard Time. So use some of that knowledge to come ask us any COVID and telehealth related questions then. Thank you to all of our speakers for sharing your knowledge and insight about all things COVID and telehealth related. And thank you all for paying attention and giving us so many knowledgeable questions. There are so many wonderful resources related to this session that can be found in the session resource page. For this session, you'll be able to find slides that we shared and other items speakers thought that you might find valuable.